Hey everyone, we are going to continue our trip through the digestive system. Last lesson we talked about the mouth and stomach specifically. Today we move into sort of the latter parts, which are the small intestine, large intestine, and then ultimately how the waste leaves the body. So far, just to recap, we've talked really about digestion starting in the mouth and stomach. So far, we have not actually absorbed any of the nutrients. We've only just scratched the surface. We've only started to digest carbohydrates and proteins. We haven't even gotten to fats at all yet up until this point. So today's lesson, we are going to cover some details about how the digestive process continues, but also then how does those nutrients actually get absorbed into the body. Because as a reminder, the whole goal of the digestive system is to make accessible those nutrients from our food and make those accessible to our cells. The goal of which being, especially, that we get glucose to our cells for cellular respiration. So we're going to continue on here talking about how that actually happens. So in the small intestine, which is the next section after the stomach, this is the location where most chemical digestion occurs, as well as absorption of most of the nutrients. So far we've seen chemical digestion in two places. One was salivary amylase, which was breaking down starches into maltose, and the second one was pepsin in the stomach, which is starting to break apart those huge proteins that we talked about. So at this point where we're just entering the small intestine, the carbohydrates are mostly digested, proteins are only partially digested, and fats have not been touched at all. So that's what we start into the small intestine looking at. So the small intestine is made of three sections. They're called the duodenum, jejunum, and ileum. And on average, just the small intestine alone is about 5 to 7 meters. So again, I gave you this sort of average amount the other day, which was like 7 to 9 meters for the entire system. So a huge majority of that is really the small intestine. It is about 2.5 centimeters in diameter. So it is a very long but very narrow tube. Okay, if you do feel up to it, you can watch my video of the fetal pig dissection. You'll see that I stretch out the small intestines. They're really, really long, especially when you compare them to other sort of tubes within the digestive system. So we're going to focus on basically the small intestine as two sections, the duodenum where we have digestion happening, and the jejunum and ileum where we have absorption happening. So as I said, the duodenum is where most chemical digestion occurs. And this is by, again, addition of those enzymes. So we're going to talk about a few enzymes here. This is not an exhaustive list. I'm just highlighting the main ones for you. But remembering that enzymes are those machinery that actually make reactions happen. There are no absorption of nutrients within the duodenum. The duodenum's focus is really just breaking things down. Okay, so we're going to see if you think back to the lesson 1, 2, and 3, we talked about all those digestive steps. I had put a diagram for you of the whole digestive system. So what I would recommend doing is sort of going back to that and sort of filling in for yourself what's happening at each place. So what I'd like to talk about here is the accessory organs. So these are organs which are contributing to digestion, but no partially digested material actually runs through them. So they are making things like enzymes, they are secreting hormones, they're doing all of that kind of stuff. And they're contributing into the small intestine, but no actual partially digested material runs through there. So the liver is extremely important. And in terms of what we're talking about with digestion, the liver is producing a substance which is called bile. And bile is actually not an enzyme. It is an emulsifying agent. And I've drawn a little picture here. So we talked previously about the fact that fat does not dissolve in water. In fact, it'll form like little droplets that won't mix into the water. So because of that, we need sort of like an additional chemical to help us just to break those big sort of droplets of fat into smaller ones. So the job of bile is to basically take these big fat droplets that are in our chyme and to separate them into smaller droplets of fat. And what this allows, and you can see in my little red picture, is it allows the enzymes to actually then access the fat from all angles. Because if you have it in a big, huge droplet, enzymes can only work on it from the outside of that droplet. So bile is not an enzyme, but it is helping with the digestive process. So what I'm showing on the side with the liver diagram, you can see the liver, and then right off the side of it is like a little sac, which is called the gallbladder. And the gallbladder and the liver are connected by ducts, which are essentially like little passageways, little tubes. But you can see here what I've drawn in the green arrows is the liver is producing bile, which is then stored in the gallbladder. Now, if you have enough already, the liver should not be producing more bile, but as you eat, it's sort of releasing it into the duodenum, into that first section after the stomach. So then we have the gallbladder, as I mentioned. This is really a storage organ. So it will contract and actually send some bile into the duodenum as needed. You might have heard that some people you know have had gallstones, which is like a little blockage that happens here. And that happens because the substance of bile itself has minerals in it. So it can actually solidify and form little crystals there. So people may have to have this removed if that becomes problematic for them. It's not an essential organ because it's not actually producing anything, but people who have had this taken out, they may have some trouble with digesting fats. So they might have to keep a bit of a lower fat diet in order to essentially ensure that their liver is able to make enough bile to be used sort of as soon as they eat because they don't have that storage organ anymore. So moving on to the pancreas, which I've also indicated on this same diagram, you'll see that nothing is actually going from the pancreas to the liver or anything like that. Pancreas is extremely, extremely important as an exocrine gland, as an endocrine gland. We talk about that next year in grade 12, but I mentioned it here. Its endocrine function is to produce hormones insulin and glucagon. So endocrine system is essentially control around the body by hormones. And you might have heard of insulin before. That's the one that allows glucose into our cells. So from the digestive system perspective, insulin is really important because, as I said before, the whole goal of the digestive system is getting glucose and other nutrients to the cells that need it. And so ultimately, insulin is important for that, and glucagon has the opposite effect, which I'm not going to go into right now, but we consider those endocrine functions. They produce hormones. So our focus actually about the pancreas this year in grade 11 is exocrine function, and that is essentially that it will make enzymes that are needed for digestion, and it will just put them directly into the duodenum, as opposed to hormones, which enter into the bloodstream. So I've mentioned here three main sort of enzymes that the pancreas is responsible for making. I mentioned before, this is not an exhaustive list. So if you Google enzymes involved in digestion, you'll find way more than this. But this is just the ones that I'm focusing on because, again, there's so many of them, we have to choose a few just to focus with. So we have pancreatic amylase. Again, it's continuing the digestion of starch because starch are these big, huge molecules. So we often have to break them into smaller parts. I told you before that it was maltose, are those double ring structures that will come from this reaction. And maltose actually has its own enzyme to then break it down, which is called maltase. And so this process goes on and on. Sucrase is an enzyme to break down sucrose. Lactase is the enzyme to break down lactose, and so on and so on. So we're not going to go into those details now. We also have trypsin. This is continuing to break down proteins. I mentioned before that the proteins are really only partially digested when you leave the stomach because they've only just started to be digested by pepsin in there. So we need more time to break those down because they're really, really huge. And last but not least, we have a group of enzymes called the lipases. Again, their job is to digest lipids or fats into those component fatty acids. So you're probably noticing that there's a lot of enzymes that end in ASE. That is like really, really common. As you see more enzymes, you'll realize how common that is. Not everyone does end in that, but if you see A's at the end, ASE, it's a good sign that it is an enzyme. So just something to keep in your mind. So a little bit of stuff about hormones. Again, we do a whole unit next year in grade 12 about this. Hormones are messengers that allow organs to communicate, and they essentially do that through communication through the bloodstream. So hormones are released in the bloodstream and they travel from one organ to the next organ. So that's one way that our bodies control what's called homeostasis, or sort of our internal environment. So hormones secreted by the duodenum control some functions of the pancreas and liver. So just thinking sensibly, in the duodenum, as soon as chyme enters there, it wants to trigger sort of the series of events that are going to bring all those good enzymes in needed to break down the food. As I mentioned with the last lesson, we don't want those enzymes constantly active, because if that were the case, it's going to cause damage to our organs. So we want to have a way to signal to turn things on and off. That's really, really important in the digestive system and also elsewhere in our bodies too. We don't want to overreact to any one thing because it pushes the homeostasis in the wrong direction. So I've listed here a couple of different hormones. CCK is secreted by the duodenum, and it's essentially, again, just signaling the pancreas to secrete certain digestive hormones and enzymes. As I said, that's typically when the chyme enters from the stomach. Then we have prosecretin and secretin, and as I mentioned in lesson 3, that's an inactive and an active form. Again, for the whole purpose that we don't want things to be active all the time, we want the ability to turn them on and off. So this one here actually relies on pH. So I mentioned previously that in the stomach, the pH is very low. It's very acidic. So when the acidic chyme enters into that section of the duodenum, that first is after the stomach, we're going to have this activation happen, and it stimulates bile to be released, and it also starts to stimulate release of lipases and trypsin enzymes from the pancreas. So essentially, the other part to this is that we don't want to keep that chyme really, really acidic. We actually want to make it closer to neutral because, as you might know, acids tend to burn tissues. And especially with a very low pH around 2 or 2.5, that's really, really acidic. So secretin is going to help with releasing some bicarbonate ions, which is going to help neutralize the pH. In doing that, you're also actually causing pepsin to become inactive, which I'm going to mention down below again. We're increasing the pH, so pepsin is going to change back into pepsinogen that we mentioned from yesterday's lesson. So secretin also has the ability to regulate the rate of digestion. So I was speaking before in lesson 3 about actually being able to close the pyloric sphincter. And secretin has a really important job with that. It's basically signaling to the pyloric sphincter, we need you to stay closed because we're not finished digesting this material. As I said before, if you aren't allowing time to digest it, it's going to just move right through your system and you're not going to absorb any of those nutrients. So you would end up actually malnourished even though you might be eating. So the body has ways to control that, making sure that the rate is not too fast. And the last one I'll mention in this section is actually an enzyme. So we have trypsinogen, the inactive form, and trypsin, the active form. And as I said, the bicarbonate ions increase the pH, which then inactivates pepsin to prevent damage. While it's doing that, it's also actually activating the enzyme called trypsin. It's going to actually continue to work on and break up those proteins into single amino acids. So what I've drawn down here is an example of a generalized amino acid. As I said previously, this would be called a monomer, which is like a single unit. In reality, proteins are like super complex. Like you could have hundreds, if not thousands of these linked together in what we call a peptide bond. So again, a big, big concept that we learn in grade 12. I'm not going to go into the reactions that involve that because that's like a biochemistry concept. But essentially these small units, these amino acids, we're able to actually absorb those into our bloodstream. The really large ones that are the proteins, which is all of them linked together, we can't absorb those. It's way too big. It won't actually enter into the blood. So that brings me to basically the end of digestion. So this is now the chyme has moved all the way through this super long duodenum. Really now at this point, your digestion process is over. And now what you're starting to do is pull those nutrients in to actually absorb those nutrients. And as I said, that happens in the jejunum and ileum. There's really no distinguishing between the two of them. They sort of flow into each other. So now we have these individual pieces, these individual monomers. And what I'm showing down here is if we were to look at the interior of the jejunum and ileum. And what we see is that it's highly folded, okay? And if you zoom in even smaller onto the folds, you see these, they look like fingers. They are called villi. And if you zoom even further onto those, you see even smaller ones, which are called microvilli. They're like little hairs. And the goal of both the folds, the villi, and the microvilli is to increase surface area. So I've mentioned previously that an increased surface area for a cell is the most effective and most efficient for absorption, specifically absorption of nutrients. So that's one of the reasons why our cell cannot be so big. So the same thing is true here. As we increase your surface area, we also end up increasing ability or effectiveness of absorption. And the whole goal of this whole section is just to absorb nutrients. So I want to just speak a little bit to this picture. Within our villi, we see capillaries, which are blood vessels, and right in the center, you will see the lacteal. The lacteal is sort of like a yellowish color in this picture. It belongs to the immune system, which is a lymphatic vessel essentially. And through that lacteal, you will actually absorb things like fats, as well as anything that is hydrophobic, so anything that doesn't like to be dissolved in water. So a little bit more about that after, which is right here. Any of your water-soluble nutrients, which can dissolve in water, can be absorbed into the bloodstream. So that's through the capillary network. Again, capillaries are super, super small, narrow vessels. They have an extremely high surface area. And as I mentioned, which I'm going to talk about in lesson 5 as well, that small surface or large surface area, small diameter, is really what allows them to efficiently exchange oxygen and carbon dioxide. So anything that's water soluble, such as the carbs, certain amino acids, other things as well, they are absorbed directly into the bloodstream. The non-polar things, such as the fats, as well as some amino acids too, actually, they have to enter through the lacteal, and they will be processed over many steps until they can actually be put into the bloodstream. So again, we don't really talk about that this year. Next year, we will talk a lot more about the chemistry behind that. It's just to say that not everything is absorbed into the blood. Some things have to be processed ahead of that. So next we'll talk about movement of the nutrients. These are a few different ways that things can get into cells. And in the example we're talking about, it's how do things get into not only the cell, but into the actual body. So from the inside of that tube, inside of the small intestine, how do they actually get into the bloodstream? So again, we do a whole big lesson about this next year in grade 12, but just for now, we have 2 types of transport that can happen: passive transport and active transport. Passive transport relies on the fact that things move from a high to a low concentration. So over here, I've put extracellular fluid, but let's say, for example, that it was in the small intestine, on the inside of the small intestine. Well, it's basically a high concentration area. It's a place where nutrients have just been released from their various foods, and so our body can pull them in. So passive transport relies on essentially moving things from a high to low concentration. In active transport, we have the expenditure or the spending of energy. And I mentioned here it's in the form of ATP, which is cellular energy. I mentioned that in a prior lesson. And this is also very selective. So in both scenarios, it's not that just anything can get through these barriers. It's very specific. So you'll notice that those purple transport proteins, they have really specific shapes That's actually allowing them to move things through or act as a tunnel. Even with diffusion, which is way over here, and I mentioned it below, we can have passage of molecules directly through the membrane without any assistance, without any facilitation. However, it's only going to be very small things that can fit through this way, as well as things that are non-polar. So again, big topic of grade 12 bio. We're not going to touch upon why those are, but later you will talk more about them, I promise. In facilitated, we rely on the proteins, okay, but we're still moving stuff from high to low. So again, active transport, we're moving in the opposite direction, a low to high. These are always going to require energy, which is ATP, and these are always going to require help from a membrane protein. So for the most part, we are relying on passive transport in the digestive system, but sometimes active transport as well. It depends what nutrients that we're looking at. So next and final section, the large intestine. So the reason we call it large is not because the length is long, but because the diameter is very wide. So it's 7.5 centimeters in diameter, and the length is fairly short as compared to what we talked about in the small intestine. It's only about 1 to 2 meters in length. So here there are a few really important things that happen. Number one is absorption of vitamins. And the reason this happens here, and I've mentioned it down below, the colon and some of this large intestine, they contain over 500 bacterial species. They digest things that we humans cannot. We don't have the nutrients for them. And so what these bacteria are essentially doing, they're living symbiotically with us. They're breaking down all of these really good vitamins and minerals, and they're releasing it so that we can actually absorb it into the body. So things like certain vitamins that I've mentioned there, certain minerals as well, water for sure, those are all absorbed in the large intestine. Now, I just wanted to mention really briefly here about the water before I go on. Water is extremely, extremely important. We need there to be water dissolving this partially digested food. At this point, we're going to start calling it waste because now after all these things have been taken out, what's left over is stuff that the body doesn't want anymore. So it's really important that there's water there just for digestive health, just to make sure that actually everything gets from point A to point B. So if you are extremely dehydrated, you also could end up constipated, which is you basically have, you're not having the flow of waste out anymore, which is really not good for anyone. Also, just to mention here, you'll see the appendix. It's right at like the beginning of the colon, the beginning of the large intestine. And I mentioned previously, this is a vestigial feature. It might have helped with digestion in our ancestors, but currently it has no function that we now currently see on Earth at all. Okay. And the final section is the rectum and the anus. The job of cellulose, which is like dietary fiber, is to help us keep full, number one, and also help to hold on to water. So it basically allows there to be some water still in the waste at the very end, so the body can't take all of it out. As I said before, that helps us to push the waste through. The waste is stored in the rectum and then ultimately until it's time to eliminate it. So you'll see at the end here, there is a set of sphincters. There is an internal and an external. The internal says that it is a smooth muscle. That means that it's controlled involuntarily. The external sphincter is a skeletal muscle. That means that we have conscious control over it. So once we are sort of like potty trained, so to speak, we start to have control over that and we understand when we have to go relieve the waste or when we don't have to do that. And I've mentioned right here, there are nerves throughout this entire rectum area, which is essentially communicating with the brain, when do you have to actually go, okay? So again, when the sphincters relax, which we've seen sphincters elsewhere relaxing as well, the final waste, also called the feces, is going to be released from the body. At the very end here, we have just the stages of the digestive process here. So those stages that I was mentioning, those are really going to include things like